My name is Bridget, and I studied the effects of muscle fatigue from a sustained isometric contraction on EMG amplitude. EMG represents muscle activation, and it can be used to detect muscle fatigue. Studies show that amplitude tends to increase over time during sustained submaximal isometric contractions. Some studies have shown a linear increase, while others have shown a nonlinear increase. So the purpose of my study was to determine the shape of the EMG curve for wrist flexors and extensors as the amplitude increases to the point of fatigue. My hypothesis is that EMG amplitude for both the flexors and extensors will increase parabolically over the course of the sustained isometric contraction. I included 8 subjects, 75% male and 25% female, and they were first-year students of the Regis DPT class. I excluded those who participated in 2 of the other lab activities that day and who had an injured forearm, hand, or fingers. For our methods, we placed 2 electrodes on the wrist flexors, 2 on the extensors, and 2 on the grounding muscles of the dominant arm. For part 1, the subjects performed a maximum voluntary contraction for 3 to 5 seconds using a hand dynamometer in standard position. The raters recorded the peak force and the peak EMG for their flexors and extensors. For the fatigue test, subjects gripped the dynamometer with 25% of their MVC until voluntary fatigue or for a maximum of 5 minutes. The raters recorded the force and peak EMG for the flexors and extensors every 15 seconds, and then they recorded the total time. For statistical analysis, we calculated percent standard and calculated the mean and standard deviation of the percent standard for force and the EMG of flexors and extensors. The graph shows mean percent standard versus time, and if you look at the blue line, that represents the EMG for flexors, which was relatively constant for the first 3 minutes, then decreased rapidly and was relatively constant after that. The yellow line shows EMG for extensors, and that was relatively constant throughout, although it had some fluctuations. And the gray line shows the force output, which was relatively constant throughout, which is what we had hoped for. Our results did not support our hypothesis, since the amplitude did not increase for either the flexors or extensors. This could be due to the fact that subjects may not have reached their fatigue point, since 5 of them lasted for the entire 5 minutes. Previous studies showed mixed results, with the amplitude increasing for certain subjects but decreasing for others as they reached their fatigue point. To measure the effects of fatigue on our patients, we must ensure that they have reached their fatigue point, and we should also note that EMG amplitude may not be the most reliable indicator of fatigue.